The next session is about finding sustainable sources of plant-based foods. And we'll start with Assistant Professor Ditte Baun Hermund from the research group on bioactives. Ditte, yeah, you're over here. And you're going to talk about plant-based seafood, algae as a source of functional ingredients and nutrients. Please take the floor.
Thank you. Okay, so I guess this is my slides. Thank you for inviting me to speak today about plant-based seafood and gave me the opportunity to talk about seaweed and microalgae. So the reason why we look at microalgae and seaweed as something related to seafood is because it comes from the ocean or from something related to marine, and also because algae actually contains a lot of the same nutrients that seafood does, like fish and shellfish as we know it. Yeah, and I'm part of this research group where we do a lot of this knowledge gaps closing by taking up some of the problematic things on producing these new foods based on algae. So we all eat food. It's kind of a fuel. We need it to get our body to function well. So in Denmark, we have these recommendations that we are trying to eat after that we need to have a protein sources. The new recommendations are that we need to eat more of the legume and these chickpeas and stuff, and less of the very fatty or saturated fat in our protein sources, such as the red meat. But we also need to eat a lot of vegetables to have a longer life, as Olivier also talked about. And we need to have this carbohydrate coming from, for example, here we have these whole grain products. But where does microalgae and seaweed actually fit into this kind of recommendations? Because there are nothing there. But the algae as a whole has a high content of omega-3 fatty acids and also omega-6, which we typically find in these seafood products. We also see microalgae that are high in vitamin D, and especially the vitamin D3, which is something that we have looked into, which you normally find in the fatty fish like salmon. It's also high in the essential amino acids and proteins, which makes it a really good source of something which is comparable to what we know from animal products. But it's also very high in vitamins. Like I mentioned the vitamin D, but it actually also contains these vitamin E's, and some of them also contains vitamin C. Then it's also very high in dietary fibers, especially the seaweed. We have a lot of these non-digestible fibers that are good for the microbes in the gut. And then we actually also have, like seaweed is low in fat, or the fat that is there is high in unsaturated fatty acids. The same goes for the microalgae, but there you are quite higher in the fat content. They are low in the sugars. Here I'm talking about the refined sugars, the monosaccharides. And it's also low in the salt. Not that it's low in salt, but it's low in the salt as we know it from the table salt. But it's actually high in potassium chloride, which is also has some health benefits. So I'm thinking we can put here algae. Here I just gave 2 examples. We have brown, and then we have the microalgae powder. Of course, there are a lot of different varieties of seaweed and microalgae. But when we are introducing new foods, we also need to consider the taste, the safety, and the stability of the products. So first I will talk about seaweed, and I will come into microalgae later. So seaweed as a food is there's a huge variety. Here I have shown some examples of the different types of seaweed. We're talking about 3 different types. There are the browns, the greens, and the reds. And these are examples that are all from Danish waters. We have two browns. That is the Saccharina latissima, also known as the sugar kelp. And we have the bladderwrack with Fucus vesiculosus. And then we have the green in the top here, ulva, also called sea lettuce. And then Palmyra palmata. And as you can see here, here's a list of like the lipid, protein, and carbohydrate content. And you might notice that it varies a lot. And that's something that we need to consider when having seaweed as a food, that depending on season, depending on location where you're cultivating it, it's a huge variety of the different compounds that you will find. So you need to consider that when making foods, that if you want high protein, you might harvest or cultivate it somewhere where it will produce a high amount of protein. So going more into like the production of seaweed, we have deployment and harvesting, or cultivation and harvesting. And after harvesting, we have the processing. And we talk about, what I will talk about today is more on the storage stability of the products, because that's very important. As soon as you take the seaweed up from the ocean, it will start to degrade. And even within 7 days, you will have something that you can't eat anymore. It will smell horrible, and it will have lost its nutritional value and not really like to eat it. So what we're looking into is this post-harvest processing. And that is something to stabilize the biomass without compromising the nutritional value. And that could be something like drying or fermentation. And I will give an example here. So in a study, we looked at the drying of 2 different types of seaweed. We had the brown one, which is a bladderwrack, and we had the sea lettuce, the green one. Both harvested here in Denmark. And then we looked at how to stabilize the product by drying it. And we used 3 different methods to see how did it affect the sensory properties of the final product, and how does it affect like the physicochemical properties and also the nutritional value, were there any differences. And already here on the appearance, it was clear that using something like convection drying, where you use hot air and a constant flow, you could see that the color of the product was much darker. And especially for the sea lettuce, we could see that it was all curled up like in small lumps, where it was more fluffy when you used the freeze drying and the microwave vacuum drying. And this study here was actually part of a company wanting to invest in new drying facilities or equipment. And we searched for this InnoBooster from the Innovation Fund Denmark, and supported like their investment by having this study of their products. So this was kind of the visual thing. And then we looked into the nutritional composition, and we looked into the different main components of the seaweed. Especially you can see here for the bladderwrack, by drying with the 3 different types of drying methods, we saw that we had more lipids when we dried it with the convection drying and the vacuum drying. And you can't have just more lipids. The amount of fat in the product will be the same. But what we were thinking was that we actually got better opening of the cells, so the structure of the seaweed is quite rigid. And if you want to have more access to the compounds, something like vacuum and microwaves, and also the convection drying where you use this heat, could actually open the cells more. And that was something that could be related to that. Okay, when you then eat it as a whole food, you might have better access. The accessibility of the compounds will then be better for you to take up in your body. And we kind of saw the same for the lipids, for the sea lettuce, but only for when you use the vacuum drying technique. Yeah, so overall, in conclusion, in this study, we saw that we should actually consider differentiating the drying techniques depending on what type of seaweed. So it's like comparing fruit and vegetables, bananas and broccoli, you cannot really just say you can use this drying method for all your seaweed and with these conditions. Then we have, in a former PhD study, Cecilia looked at food safety. So the thing is, with something like this sugar kelp, it contains a lot of iodine. And if you have an excessive amount of iodine in your diet, you will have some health issues. So we were looking into that the fresh biomass actually contained, like what we can see is up to, if you have it in a wet form, it was up to 2.5 times more than what we recommend for the daily intake of iodine, and 10 times higher if you dried it. So what our PhD study was considering was, okay, how do we get rid of the iodine? So the iodine is water soluble. So we were thinking by blanching it, like you do with vegetables, and some vegetables actually need to be blanched, like some beans and also potatoes, to make it eatable. So we did like a study where we blanched the seaweed at different temperatures, and saw how time affected the iodine reduction. And we could see that we could get a quite high reduction by just dipping it for like 5 seconds in 80 degrees hot water. And you can see here also how it changes the color of the seaweed and actually makes it more appetizing to eat. Okay, to microalgae. The production of microalgae is like you have a media where you then add your microalgae culture, and then it will like multiply. It will not grow in size, it will multiply into it has a density where it can't grow anymore, and then you harvest it. So here you see what is bubbling is like a lab scale. We also have that at our facilities, and this afternoon Emil will give a tour to the algae lab where you can see something like this. And then we have more like a pilot scale where you have tubes where you're under constant turbulent flow will have your cultivation of your microalgae. And this is just an example of how you can grow the microalgae phototrophically. That means that they need light, they need air, and they need this media. And then after we have cultivated and you go to a certain density where they don't grow anymore, you can you harvest them, you concentrate them into like a paste, like if you imagine like some slurry kind of thing with a dry matter of approximately 30%. And then from there, you can dry it. You normally also have a homogenization step where you can break the cells, because the same as for the seaweed, you need to open the cells to get access to the good stuff. And then you end up with something like this. This is an example of a chlorella powder, where you can actually have up to 60% protein in this dry powder. And then to the application, which we have worked on, you can either do some more processing and take out the oil. Microalgae can contain up to 50% oil, which is a really good source of these omega-3 fatty acids. And you can also use it directly as a supplement to a food. In this case, we made protein-rich bread. So the focus that we have been doing is how do we cultivate the seaweed? Can we cultivate, for example, of something like industrial water? Can we optimize the nutrients? How do we do the concentrating and the drying of the microalgae? And also what could the applications be, and what is the accessibility? Like how well are the compounds inside the seaweed actually, how accessible are they? And that is directly linked to some of the projects that have been here at my research group for the past years, where we have done studies of this. And right now we have the mass project ongoing, where we're trying to optimize the production of Nanochloropsis oceanica. which is very interesting, and I will show you that in the next slide. Because Nanocoropsis oceanica has the ability to actually produce or synthesize vitamin D3. And that is if we expose it to UVB. So when we have the live culture, you can add UVB radiation, and you will get it to synthesize these vitamin D3. So in a study, in one of these projects, the UVB radiation were done at different times of the process. In the live culture, in this slurry that I talked about, this 30% dry matter thing, and in the dried powder. And it was clear that the vitamin D production was best when you did it on the live culture. So we managed to get going from the non-treated one with a lower than 1 nanogram per gram dry weight up to 285 nanograms per gram dry weight in the final dried powder. And just to give an example of what is actually found in the salmon, we have something like up to 185 nanograms per gram of salmon. So it's quite high. Then to the formulation of the microalgae, if we were to add it to these protein-enriched bread. So first of all, you can see that it's very green. Here we added in, it was 3 different concentrations of 2 types of microalgae, both green microalgae, and it added this color, which might not be that appealing if you were going to eat it. But the other thing was also that you added a lot of taste that was not really associated to eating bread. Besides the green color, you actually had bitterness. And the bitterness is something that we often see in these microalgae because it contains compounds that are bitter. But it also has a seaweed odor and a seaweed taste, which also is associated both to these omega-3 fatty acids, of course. If they go rancid, you will have the taste of fishy off flavors, which is something that is not really nice to have in a food product. And then the saltiness. So I think that if you take this experience into consideration, you might think of more applications that are more related to seafood applications in the future. So just to sum up, seaweed and microalgae as a future food, how do we think about it? So we have looked into this about the cultivation and what are we doing with the processing, but also how do we stabilize the product by, for example, drying it or doing some fermentation or some further processing. And then, of course, the food safety. So it's rich in protein, amino acids, omega-3 fatty acids, vitamins, and minerals. But you can also actually take compounds out of it, like extraction. We do different extraction techniques, for example, the supercritical CO2 extraction, which is really popular at the moment, where you can take out different compounds like antioxidants or use it for stabilizers. And then when it comes down to taste, always when you're introducing new foods, if you want to know more, we have some taste samples in the afternoon in our food fair, where you can come and taste some microalgae products. Thank you.
Thank you, Ditte.